Welcome! Today we're going to explore restriction mapping. There are two main types of DNA mapping. Genetic mapping, based on how often genes are inherited together. It gives the relative positions of genes using units called centimorgans. Physical mapping provides the actual physical locations of genes or markers on the DNA strand, measured in base pairs. Restriction mapping falls under physical mapping, and it uses special enzymes to cut DNA and map the cut sites. By cutting DNA with one or more enzymes and measuring the sizes of the resulting fragments, we can figure out where the cuts occurred and draw a restriction map. In linear restriction mapping, we work with a straight DNA strand, like a thread. Each enzyme cut produces fragments, which we analyze using gel electrophoresis. First, we digest the DNA with enzyme A alone. It produces two fragments. One is 10 K base pairs, and the other is 7 kilo base pairs. This means enzyme A cuts the DNA at a single site. Next, we digest the same DNA with enzyme B. This time, it cuts at two places and produces three fragments: 3 kilobase, 8 kilobase, and 6 kilobase. We digest the DNA with both enzyme A and enzyme B together. This triple cut produces four fragments: 8 kb, 6 kb, 2 kb, and 1 kb. Using the sizes of these fragments, we can piece together the exact order of the cuts along the DNA. Let's make the restriction map enzyme A and produce two fragments of 7 and 10 kb after that enzyme B cut at left and right sides of enzyme, and now we get four fragments of 6, 1, 2, and 8 kb 1 and 2 kb together make 3 kb. This arrangement satisfy all the fragments. This is the correct restriction map. Plasmids are circular DNA molecules found in bacteria. Restriction mapping works a little differently here. With one enzyme cut, the plasmid becomes a linear piece, giving one fragment. With two enzymes, we get two or more fragments depending on their positions. Example, enzyme A produce 1 6 kb fragment, and enzymes A plus B produce two fragments, 2 kb and 4 kb. Application and significance of restriction mapping. So, why is restriction mapping important? It's a fundamental tool in molecular biology with wide-ranging applications, including gene cloning, locating insertion points for genes of interest; genetic engineering, modifying plasmids for recombinant DNA; diagnostic testing, detecting mutations or deletions in DNA; forensic science, analyzing DNA patterns in criminal investigations; genome research, constructing physical maps before full genome sequencing. Before modern sequencing methods, restriction mapping was the go-to method for understanding DNA structure. And even today, it remains a valuable technique for quick and cost-effective DNA analysis. Restriction mapping paved the way for biotechnology, medicine, and genetic research as we know them.
Please like, share, and subscribe, and turn the bell icon for more videos.